Okay, can somebody confirm you can hear me?
Yes.
Yes.
Thank you. All right, before we begin, are there any questions? What's going on with Zoom here? Oh, maybe the lecture file is finished. No, it's 95% done, so not sure why it was flashing. All right, let's begin. Syllabus here. Let me share my screen. So many windows, I'm having a hard time figuring out which one to go to. There we go. Oops, 97%. That's the lecture file is being made. I don't know why that came up here. Let's see if I can get rid of it. All right, today is April 23rd. And on this week, we only have one lab, Lab 4, which we'll discuss today. And then on Thursday, there is no lab. Whenever there's a day when there's no lab, I will show up to answer questions, and I will be here for the first 15 minutes of the lab. And then after that, I will log off. So if you have questions or you want to talk to me, you can log in on those days at the first 15 minutes of the lab, and I'll be there to answer questions or to talk to you. But other than that, there will be no lab on Thursday. And if you have no questions, you do not need to log in. Any questions about any of that? What you should be doing on a day like this is working on your infectious disease project. And later in the term, you should be working on your unknown work or your infectious disease project. So this is the first time we've had it this year, but we will have it again. Well, in week 6. In week 7, you really won't have it because I got to discuss the unknown project. And then we're doing one lab. So we'll be discussing the unknown project in one day and the lab on another day. But on week 6, we'll only have one lab. Like week 3, only one lab. Lab 4 is going to cover stains. Any questions about what we're doing? If not, I'm going to go to lab 4. So there is some reading in the textbook as normal. And sorry, this is a weird color, but I don't know what got into my computer, but it's showing things with a very strange background, and then the text is white instead of the background. Usually the text is dark and the background is light, and then the highlighting is really funny. But you should read about size, shape, and arrangement of bacterial cells, 2 pages. You should read about the bacterial cell wall, 3 pages. You should read about the simple stain, just a page or less. You should read about differential stains, about 2 pages. It'll include the gram stain and the acid-fast stain. We'll talk about both of those stains today. And then you should read about special stains, a page to maybe 2 pages. We'll talk about negative staining to see cell capsules. We'll look at endospore staining and flagella staining when we're talking about special stains. There are some video clips in a virtual lab in today's lab. The learning objectives: 1, be able to prepare a bacterial smear for staining. And let me reiterate that when you make a bacterial smear, it is something you do before you do any staining. Any questions about any of that? All right, because always somebody gets a question wrong on the quiz about bacteria smears. Every term I've been teaching. I don't know what it is. They're just not paying attention to me or to the lesson, and they just go on and do something. But anyways, primary learning objective 2, understand the principles and the procedures used when performing a simple stain, a differential stain, and a special stain. 3, interpret the results for a simple stain, a differential stain, and a special stain. And then be able to define the following terms. What's a simple stain, a differential stain, a special stain? What's a coccus shell cell, a bacillus cell, a spirillum? What's peptidoglycan? What's the gram-positive stain? What's the gram-negative stain? What is an acid-fast? And when I say gram-positive stain, I'm talking about a cell, because there is no gram-positive stain or a gram-negative stain. It's the gram stain. But the cell stains either gram-positive or gram-negative. What's the acid-fast stain? What's the primary stain? What is a mordant? What is a decolorizer? What is the counter stain? What is a capsule? What is an endospore? And what is a flagella? Any questions about what we're going to be covering? All right. Because bacteria are essentially transparent, it is necessary to stain them before they can be observed under the microscope. When we're going to do this, we first make a bacterial smear. You add a drop of sterile water on a clean slide. You then add the bacteria. The bacteria are mixed in the drop of water, and then the smear, the bacteria and the water, are smeared over the surface of the slide. The slide is then allowed to air dry, and then the organisms are fixed onto the slide by gentle heating. We usually call that heat fixing. Because of the air drying and the heat fixing, the cells are then not washed off during the staining process. Simply adding the stain will be in a liquid, usually water, and that can wash away the cells if they're not fixed to the slide. A simple staining technique uses one dye to stain the bacterial cell. You'll note that the lesson used to say a basic dye. That's not correct. If you're using one acidic dye, it's still a simple stain.
Aniki, will you come here?
Okay, somebody, you have your microscope on. Could you turn it off? Not your microscope, your microphone. Let me see if I can find who that is. Matthew, could you? No, it's not Matthew. They must have turned it off. So a simple stain is using one dye to stain bacterial cells. You can get the shape and the arrangement of the cells, but to get anything further, you must use a differential stain. A differential stain is a technique usually using two or more dyes to provide additional information about the cell. You can also use a special stain, and these stains are used to stain particular bacterial structures, such as the cell capsule, the endospores, or the flagella. Any question about any of that? So when we're looking at cells, you can note that they have a different shape. There's a cocci, a round type cell, and a bacilli, a rod, and then there's a spiral, an S-type shape. But there's also growth arrangements. With the cocci, you can have singlets, which isn't shown here. You can have diplococci, shown here. You can have tetrads, shown here. You can have I'm losing the name of that. Sarcenia, which is 8 cells stuck together. You can have staphylococci, and that's just a clump of cocci. And you can have streptococci, where the cells are hooked together in a row. Streptococci. Any question about that? Now, with the cocci, you have more cellular growth arrangements than you do for the bacilli or for the spirilla. For the bacilli, you can have singlets, shown here. You can have diplobacilli. It's not really shown, but two bacilli hooked together like the cocci. And then you can have streptobacilli, where the bacilli are linked together in a line, and they're linked together lengthwise. Let me draw that. So streptobacilli is where the cells are hooked together this way. Any questions? You can also have the cells hooked together this way. I have to erase that. And this is not called streptobacilli. What does this kind of look like? This is called a palisade. I'll put it down here. That's not spelled right. Palisade. This is a palisade fence where you get a board, piece of wood, put it in that way, and hook it to the wood that way and that way. And I know palisade fences aren't very common anymore, but in Mark Twain, they were very common in his days. And I think it was Mark Twain was painting a palisade fence. And not Mark Twain, it was Tom Sawyer who was painting the palisade fence. And he told Huckleberry Finn that it was really fun to do and got Huckleberry Finn to paint the fence for him. But anyways, the fence they did was a palisade fence. And it's shaped like this, and that's where the name palisade comes from. Any question about any of that? And that's where they're stuck on the this edge and that edge, as opposed to this edge and that edge. The important part is that the bacilli have less cellular growth arrangements than the cocci. And then the spirilla only have singlets. That's the only cellular growth arrangement the spirilla have. Any question about any of them? All right, let's talk about differential stains. There are two. The first is the gram stain. And if ever you're using the name Gram, it should be capitalized with a capital G, because Graham is named after a person, Hans Christian Graham, who was a doctor. You don't need to know the year, but he was trying to come up with a stain to differentiate between the bacteria of the infection and the patient's own cells. And so his stain was differential, it stained the bacteria a purple, and then the patient's own cells were stained a pinkish color. And then biologists started using Graham's stain, and they came to realize that it was differential among bacteria, separating bacteria into two very large groups of cells. The gram-positive bacteria, which stain purple, and the gram-negative bacteria, which stain pink. And these two cells differ in their cell wall. We'll talk more about the differences in a little bit. But here you can see gram-negative bacillus, a gram-negative spirillum, a gram-positive bacillus, a gram-negative cocci. I don't see anything else. The gram stain distinguishes between different bacteria according to their ability to retain the certain dye. Specifically, the crystal violet is retained by the bacteria if the bacteria is gram-positive, and these cells will stain purple. If, on the other hand, the cells lose the crystal violet, they do not retain it, then they are called gram-negative, and they will appear pinkish in color. Any question about any of that? The difference is the cell wall composition between the gram-positive bacteria and the gram-negative bacteria. The gram-positive bacteria have a very thick cell wall, such as a very thick peptidoglycan, which retains the crystal violet, whereas the gram-negative bacteria have a much thinner cell wall, and they have a thin peptidoglycan layer, and it does not retain the crystal violet. Any question about any of that? So there's the gram-positive bacteria showing you the cell wall, which is cut off a little bit, but you can see it has a very thick peptidoglycan layer upon layer of peptidoglycan. Whereas the gram-negative cell wall is shown here. This is the plasma membrane. There is the peptidoglycan layer of the gram-negative cell. And then here's the outer membrane of the gram-negative cell. And the important part to recognize is this peptidoglycan layer is thinner on the gram-negative cell than on the gram-positive cell. And you can see that there are layers of peptidoglycan, but many less than the gram-positive cell. And usually the number of layers of the gram-negative cell are between 3 layers of peptidoglycan to about 20 layers of peptidoglycan. Whereas the peptidoglycan layer of the gram-positive cell is much greater than 20. It may be 100 layers or more of peptidoglycan. So in the gram stain, after bacteria are stained with crystal violet, Graham's iodine is used as a mordant. A mordant chemically combines with the first dye, the crystal violet, and forms a crystal violet iodine complex. This crystal violet iodine complex is larger than the crystal violet alone or the iodine alone. Then an alcohol acetone solution is used as a decolorizer, which is applied briefly to remove the excess crystal violet. In the gram-negative cells, the crystal violet is decolorized. It's totally washed out of the cell. In the gram-positive cells, the crystal violet is not washed out by the decolorizer, and the cells retain the crystal violet. The timing of the decolorizing step is critical. And we'll talk more about that in a little bit. But if you under decolorize, you will not remove the crystal violet from a gram-negative cell, even though it should be decolorized. And if you over decolorize, you can start removing the crystal violet from the gram-positive cells, even though it should not be removed from the gram-positive cells. So the timing of the decolorizing step is critical. And after a successful gram stain, the gram-positive cells will stain purple, the gram-negative cells will stain pinkish. How the cells stain pinkish is after decolorization, you then add the counterstain safranin, which stains the cells pink. And so the gram-negative cells will pick up the pinkish color from the safranin. So here are some cells stained with gram stain. Some of them are staining gram positive. Some of them are staining gram negative, the pinkish cells. Any question about any of this? Now, the gram stain does not stain all cells well. With the mycobacteria in particular, the mycobacteria tend to resist getting the dyes of the gram stain. And we'll talk a little bit more about that. The mycobacteria have mycolic acid in their cell wall. Mycolic acid does not allow water and substances that are dissolved in water to come into the cell, at least not easily. And so the mycobacterium usually stain lighter than other cells if the cell is gram positive. And that's because less of the crystal violet comes into the cell because of the mycolic acid, which you'll learn about later. And if you don't do the gram stain well, the cells may not even pick up the crystal violet. Sometimes the mycobacterium also stain gram variable, meaning some cells stain gram positive, other cells stain gram negative. And this is actually an example of a gram variable cell. However, what do you notice about the gram positive cells, like this cell here, and the gram negative cell? Come on, let me blow this up. Like that cell here. What do you notice about difference between this cell and that cell? We could also say this pinkish cell here and that cell there. What do you notice is different? Anyone? Anyone take a guess? There's two differences.
Looks like the pink is shorter than the dark one.
Well, that's true, but that's not what we're looking at. I'm
Oh,
sure if I were to look around, I could find a large pink cell, and there's a small purple cell. There's another difference.
Is
Okay.
it the shape
Um,
of the cells?
maybe a little bit. This cell is looks like it's broken. This cell looks mostly like a ghost, and these cells are probably dead cells, and these cells are very intensely staining. There's a very distinct boundary between the cell and the outside of the cell, meaning the cell wall is very distinct. And this cell is probably a viable cell, and this cell is clearly broken, so that's a dead cell. And this is a ghost, so it's also a dead cell. So what's happening here is these are not freshly grown cells, and you have a lot of dead cells in here. The dead cells do not stain correctly with the gram stain. And so to get the gram stain to stain correctly, as well as to get it to stain well, you actually need to use it on cells that have grown for 2 days' time or less. And if you have older cells, you should not use the gram stain. You should streak out the cells and get freshly grown cells before doing the gram stain. The only time I've ever seen gram variable cells among the mycobacterium were when a student stained cells that were 2 weeks old or older. And I looked at the cells and I said, some of these cells look dead. How old are these cells? And the student told me, oh, they're at least 2 weeks old, maybe a month old. And I said, well, that's the problem with the gram stain. You have to have freshly grown cells or the gram stain will not work correctly. And that's the tip. A tip for doing the gram stain, avoid using old cells. The cells should be only 2 days old or younger before using the gram stain. Make a thin bacterial smear. If you have several layers of cells on top of each other, it's hard to get the crystal violet all the way in to the cell on the bottom. And then when you're decolorizing, like a gram-negative cell, if you have cells stacked on top of each other, it's hard to get the crystal violet to come out of the cells way at the bottom of the cells. So you want a thin smear preparation. You should heat fix the bacteria only after the smear is air dried. Because if the smear has not air dried, you can get the water in the cell to boil, and then that will cause holes in the cell membrane and cell wall. And then avoid overheating the cells. Excess heat disrupts the cell wall, you can damage it, and then the cell may not retain the crystal violet. Obviously, do not over decolorize, do not under decolorize. Any questions about any of those tips? All right, we'll go through the gram stain in more detail. My guess in this lab, or maybe in the lecture, I don't remember. With the acid-fast stains, some bacteria have a waxy substance called mycolic acid in their cell walls. These bacteria are able to resist the acid-alcohol decolorization, and therefore they are referred to as acid-fast bacteria. The mycobacterium are the main cells, which are acid-fast, but nocardia are also acid-fast bacteria. That's another family. Initially, when you stain all cells, they all take up the primary stain, the carbonyl fusion, which is cherry red, but then the cells are decolorized with acid alcohol. And then a counter stain is applied. The counter stain is either methylene blue or brilliant green or crystal violet. And then the counter stain will be either blue, green, or purple, depending on what the counter stain is. At the end of the staining process, the acid-fast stain will stain red. The non-acid fast stain will be blue or green, depending on the counter stain that's applied. So here we have cell stain. Let me blow this up. The acid-fast bacteria are staining red from the carbonyl fusion. The non-acid fast stain is this bluish green colored cell here from probably brilliant green. All right, let's move on to talking about a special stain. There are 3 special stains we're going to talk about. The capsule stain. We move on. The endospore stain and the flagella stain. Those are the 3 special stains we're going to talk about. Remember, a special stain is just staining a specific part of the bacteria, so then you can then see it. The glycocalyx is a structure which is attached to the cell wall, and if the structure of the glycocalyx is firmly attached to the cell wall, and you can see it under the light microscope, the glycocalyx is called a capsule. That's the only glycocalyx we're going to talk about here. There is a slime layer. The slime layer is mostly water. It is not firmly attached to the cell wall. And you cannot see the slime layer under the light microscope. So we're only going to talk about the capsule. A negative stain is also called a capsule stain. It stains the cell capsule. Capsules can be associated with an organism's virulence or its pathogenicity because the capsule can protect the cell from being phagocytized, and that means that the host immune system has a much harder time getting rid of the bacteria than if the cell did not have a capsule. For example, strep throat, it is very difficult to throw off a strep throat infection because the cell has a capsule. When you grow the cells that have a capsule in a colony, the cells have a mucoid-like appearance. Capsules are not easily observed with conventional staining methods because the stain washes away the capsule. Therefore, a negative staining technique is commonly employed to demonstrate a capsule. India ink or nigrosin are commonly used, and if a capsule is present, you'll see a clear halo around the cell, which is the capsule, and this is staining with India ink, which stains the cell lightly gray, and then the background is stained very dark color, like black. And this is nigrosin, and maybe another dye as well. It stains the background a lightish color. I don't know if that's greenish gray or what. And then it stains the cell a darkish color. And then once again, the capsule is clear, forming the halo around the cell. So that's Klebsiella pneumonia here, and using India ink over here. All right, any questions about the capsule stain? If not, we'll talk about the endospore. An endospore is a metabolically inactive, hardy structure that is produced by some bacteria as a defensive strategy to allow the cell to survive unfavorable environmental conditions. So when the environment becomes detrimental, the cell will start making an endospore, and then the endospore can survive when the parent cell cannot. And then when the environment becomes favorable, the endospore will germinate and the bacteria will return to their metabolically active vegetative state, which was the parental cell. Most endospores are in 2 genera, Bacillus and Clostridium. So most bacteria that have endospores belong to a member of the family Bacillus or the member of the family Clostridium. And this table is showing you some of the pathogenic endospore-forming bacteria: Bacillus anthracis, causing anthrax; Bacillus cereus, causing food poisoning; Clostridium botulinum, causing food poisoning, aka botulism; Clostridium tetani, causing tetanus. A common name for that is lockjaw. Clostridium perfringens, causing gangrene and food poisoning. And then Clostridium difficile, causing antibiotic-associated colitis, often called C. diff, or pseudomembranous colitis. Any question about any of that? All right, to see the endospore, we use the endospore stain, and one of them is the Schaefer-Fulton endospore stain. That's the only one we'll talk about. It uses malachite green as the primary stain. And then heat is applied as a mordant. The cells are then decolorized with water, which will remove the malachite green from the vegetative cell or the periental cell, but it does not remove it from the endospore. Safranin is then applied as the counterstain to stain the unstained cells a pinkish color. And so the endospore will stain green. The parent cell or the vegetative cells will stain pinkish, which is shown here. I'll blow this up. That is an endospore inside a cell. That's why there's a little bit of pink on the left side of the cell and a larger region of pink on the right side of the cell. And then the endospore is green staining in between the two sides of the cell. That is an endospore inside a cell. When the endospore is fully mature, it'll come out of the cell, usually when the cell begins to die. And there we see a free endospore, and it is greenish in color. Any question about any of that? Any question about the endospore stain? All right, the third special stain is the flagella stain. It is a stain to specifically stain the flagella and make it appear under the light microscope. Flagella are appendages, like a whip, coming out of the bacteria, which is used for locomotion, meaning it allows the cell to have motility. If the cell is a bacteria or an archaea, the only way those cells can move is with a flagella or several flagella. However, the flagella cannot be seen under normal conditions with a microscope. Flagella is composed of flagellin, a type of protein. You do not need to know this for the quiz, but you do need to know it for the lab, to answer one question in the lab. The number and distribution of flagella on the bacterial surface are useful in identifying and classifying bacteria. You can have bacteria with one flagella on the polar end, and that we call monotrichous. You can have several flagella on one end of the cell, and this is called lophotrichous. You can have several flagella on both ends of the cell, and this is called amphitrichous. And then you can have flagella all over the cell, and that is called peritrichous. Any question about that? Because the flagella are too thin to be seen using standard staining techniques, special stains are required to view the flagella under the microscope. And usually what happens is you start staining the flagella, and then you apply a mordant, and it'll coat the flagella with many different layers until the flagella can then become visible under the light microscope. And here we're seeing the flagella stain of one species of bacteria here. You can see that this one and that one and that one are clearly peritrichous, but this one is lophotrichous. I don't see any amphitrichous. If I look really hard, I'll be able to find one with just one. Maybe that one. That looks like two. Maybe that one's only got one. Monotrichous. Now these are the same species. Why might some, let's see if I can find one with none. That one might have none. Why would some have none, some one, some two or three all on the same edge, some on both edges? I don't see any off that, at least off the top of my head. I don't see any that have on both ends. And then some flagella all over the cell. Peritrichous. Anybody have a guess? Oh, there's one that's clearly just two or three on one end. The reason why some have none, some have one, some have several on one end, and some have several on both ends, and then some, like these three, have them all over the cell, is because this cell here has not put out a flagella yet. It has not formed one yet. But once it first starts forming, it'll first have one. And then it will be called monotrichous. And then it'll put out several on one end. It'll be called lophotrichous. And then it'll put out several on both ends. It'll be called amphitrichous. And then it'll start putting out flagella everywhere, and it'll be called peritrichous. So when you're naming the flagella, you look at the highest order in the species and then say the flagella on this species is peritrichous. So you're looking for the highest order and then say that's what we have. You don't say that they're all of them, okay? And if the highest order is lophotrichous, you say that it's lophotrichous. Any question about any of that? All right, so know the terms of this lab. Let me shrink that down. In today's lab, we'll not be doing any actual microbiology because everything will be done online. But you must understand how to prepare bacterial smears for staining. You must be able to understand how to do the stain. You must understand the procedures and principles for the various staining techniques and be able to interpret the results using the microscope. So in procedure one, you're going to view a video clip on preparing a bacterial smear and then on applying heat fixing to make the cells attached to the slide. Here are the steps for preparing a bacterial smear. And then answer the question number 3 on procedure 1. What are 4 common errors when preparing a bacterial smear for making a stain. Any question about that? So answer that question, procedure 1, number 3. The questions always have a question mark after it. And then move on to procedure 2. We're looking at a simple stain. View the following clip of a simple stain. And here are the steps for using a simple stain. Adding a basic dye. After about 1 minute, gently wash off the excess dye with water. Blot the slide dry with blotting paper. Let me show you. Blotting paper is thick paper, almost like cardboard. Let's pretend this is your slide. You put the slide in the blotting paper, fold it over, and then just press down to blot the slide with the blotting paper. It's also called bibulous paper, by the way. When you're blotting the slide, you will not remove most of the cells from the slide. But like I said, you just press the paper into the slide. You do not move the slide. I don't know if you can see this or not, but you do not move the slide against the blotting paper, because that will rub off the cells. And that will remove enough of the water that you can use the microscope, and you can even use oil on the microscope because enough of the water has been removed. Any question about any of that? And then answer the question, describe the cellular morphology of E. coli, which is not shown there. Oh, there it is. Sorry, it is shown. And then Staphylococcus aureus. The morphology is the shape of the cells. I'll give you a hint. E. coli is not a spirilla. This looks like a spirilla in certain places because 2 cells are linked together. Let me move this, blow this up. So right there, that's not a spirilla, that's not a spirilla. That's actually 1 cell there, 1 cell there, and then 1 cell there. Same with that. That's not an S. That's actually 1 cell here, 1 cell there, and maybe even another cell there. What's the cell morphology of Staphylococcus aureus? Write it here. What's the total magnification used for this procedure? Just go look up. And then move on to procedure 3, the gram stain. View the following clip going over the gram stain and its procedure. Then practice using or performing the gram stain using the virtual lab. When you're opening the virtual lab, it may take some time to download depending on how quick your internet connection is, how fast the web is at the moment. Go through the procedure. I'm just going to move.
Hi, Lab Assistant. All yogurt. Unfortunately, this is what plain. Now take a look at this yogurt. This yogurt looks suspicious, but to tell if it is contaminated, we need to get a much closer look. In the gram staining process, bacteria that stain a pink, we are going to gram stain. All right, let's prepare our slides for gram staining. To prevent bacteria from contaminating our work area, you're going to use a striker to light the Bunsen burner. The gas is turned on low already, so bring the striker to the Bunsen burner with the flint side facing down and squeeze the striker to create sparks.
The arrow is telling what to do. So you grab the striker and bring it down here.
We need to dilute the yogurt before we put it on the microscope slide. Start by placing 1 milliliter of the diluent into a test tube using a pipette. This is an inoculation loop, a small loop of wire used to collect samples. We'll use it to place a small amount of yogurt into the test tube with the diluent. Start by sterilizing the inoculation loop in the flame of the Bunsen burner. Hold it directly in the flame until it glows red hot. After it has cooled down, use the loop to obtain a small sample of yogurt from the suspect container. Place the yogurt sample into the previously measured diluent, PBS. Now, gently shake the test tube for 5 to 10 seconds. Use the inoculation loop to transfer a small amount of sample from the test tube to sterilize the inoculating loop again in the flame of the Bunsen burner. Hold it directly. Transfer a small amount of sample. Place 2 loops full from the sample onto the slide. Tilt the slide to spread the drop out slightly, about the size of a... Now we'll heat fix the slide. Place the slide in a clasp, such as a clothespin, and pass it over the flame of the Bunsen burner. Be careful not to put it directly in the flame. Now we begin the staining process. After the staining is complete, we can look at these slides using a microscope. We use 4 chemicals to stain a slide. We apply them in a specific order and rinse the slide off under slow running water in between each chemical. The first is crystal violet. This is our purple stain, which will indicate gram-positive bacteria. Using the dropper, squeeze out enough drops to cover your sample. Let that sit for 60 seconds.
Okay, that's starting the gram stain, so go through the gram stain procedure. And that's the virtual lab for you to actually do the gram stain. Any question about that? So make sure you do the gram stain. I'm going to continue because I don't need to go through the entire virtual lab. I know how to do the gram stain, but you guys need to go through that. And then review the steps used when performing a gram stain. First, you flood the smear with crystal violet. You let the crystal violet soak on the cells for 1 minute. Then you dump the crystal violet off and rinse the slide with distilled water. All the cells after the crystal violet will stain purple, both the gram negative and the gram positive. You then dump the water off and flood the slide with grams iodine. Grams iodine is a mordant. It will chemically complex with the crystal violet, making a larger molecule than either the crystal violet or the iodine alone. So the cells will stain a little darker and a little more purple, meaning the crystal violet is a blue purple, the grams iodine crystal violet complex is a purple color. And these cells may stain a little darker too than those cells. You then let the iodine sit on the slide for 1 minute. Dump the iodine off, rinse the slide with distilled water, and then add decolorizer. The best thing to do when you're adding the decolorizer is take the slide, put it next to your eyes, get the decolorizer, and then drop the decolorizer so that it runs down the slide. Once the blue stops running, stop adding decolorizer. If the decolorizer is done correctly, you will wash crystal violet out of the gram negative cells, but you will not wash crystal violet out of the gram positive cells. Now, like I said, that is if the gram stain is done correctly. The decolorizing step is actually the, what's that word, this step? The differential step of the gram stain, because at this point, the gram positive cells will stain purple and the gram negative cells will stain clear. So the decolorizing step is the differential step in the gram stain. You then apply safranin for 1 minute. Safranin is a pink dye. the pink dye will get in the uncolorized cells and then colorize them pink. Then you rinse, dump the saffron off, rinse the slide with distilled water, and then you blot dry the slide, and you may with bibulous paper, and then you may immediately look at it under the microscope, even if you use oil. Any question about any of that? So here are two smears using the oil objective lens with the gram stain, and then fill in the chart. E. coli is either gram positive or gram negative, so put your answer there. And Staphylococcus aureus is either gram positive or gram negative, put your answer here. E. coli, give the cellular morphology here. And then Staphylococcus aureus, give the cellular morphology here. Any question about any of that? Remember, cellular morphology is the shape of the cell. I'm not asking for the cellular growth arrangement. Any question about any of that? All right. Then for each of the following organisms, predict the color you would see, pink, purple, or colorless, after this step of the gram stain. So A, Staphylococcus aureus, after the primary stain is added, will be what color? E. coli, after the primary stain is added, will be what color? Staphylococcus aureus, after the alcohol acetone step, is going to be what color? E. coli, after the alcohol acetone step, will be what color? And then Staphylococcus aureus, after the Canner stain is added, will be what color? And E. coli, after the Canner stain is added, will be what color? Any question about any of that?
I have a question about that. So each of that, like in A, so we need three answers for these three colors, like pink, purple, and colorless.
No, and Staphylococcus aureus will be only one color after the primary stain is added.
Ah,
So you have to take pink, purple, or colorless for the possibilities.
so we choose what color. All right.
All right. In procedure 4, we're going to talk about the acid-fast stain. Here's a video clip on performing the acid-fast stain. Review the steps when performing the modified Kenyan acid-fast stain. First, you apply carbol fusion. Carbol fusion is the primary stain. You then will apply the acid alcohol wash. That will be the decolorizer. The decolorizer will pull out carbol fusion from any cell that lacks endospores, but it will not pull out the carbol fusion from the endospore or a cell containing an endospore. And then we apply brilliant green as the counter stain, just to stain the uncolored cells something so we can make them more visible. And then the acid-fast negative cells will be staining green from the brilliant green. And then the endospores, or the cells containing endospores, will, at least the endopore portion, will retain the carbol fusion, and they will stain reddish. You add carbol fusion to a heat-fixed slide, cool the bacteria. After about 4 minutes, you add the carbol fusion, and then after 4 minutes, you gently wash off the carbonyl fusion with water, shake off excess water, and then you begin the decolorization with acid alcohol for 10 to 15 seconds until you no longer see pink freely running off the smear. You then gently wash with tap water to remove the to remove the acid alcohol and prevent it from working further. You cannot over decolorize organisms that are acid fast bacteria positive. Add the counter stain, brilliant green or methylene blue, for about 1 minute. Gently rinse with water, shake off excess water, and blot dry the slide with bibulous paper. Seen are 2 smears below. Interpret whether the organisms denoted by the arrows are acid fast positive or acid fast bacteria negative. So you must, for each arrow, state either acid fast bacteria or acid fast bacteria AFB negative. State that for each arrow. Procedure 5 is looking at special stains. View the following video clips on the endospore stain and a negative stain to view the capsule. Below are 3 bacterial smears that are being observed under 1000x with oil. In figure A, with question A, this is procedure 5A. This is the Schaefer-Fulton stain, and it uses which dye as the primary stain and ultimately dyes what cell or cell structure, which are produced by some bacteria to aid in their survival. And then 1B lists some examples of pathogenic endospore producing bacteria. You can get this from the lab module. I actually went over it in class. Okay, procedure 5B. This is what stain that coats long whip-like appendages which aid in motility. And this is looking at this slide B here, procedure 5B. And then state the flagella arrangement of the types seen. Remember, you only put 1 cellular growth arrangement here. And then procedure 5B3 lists some examples of clinically significant bacteria that produce flagella. And that is discussed in the lab module. You could also go online to answer that. Procedure 5C, we're looking at what stain. It does use India ink or nigrosin and can be used to detect the presence of what cell structure, which is located exterior to the cell wall and prevents phagocytosis. So here we have it here. And then answer procedure 5C3 lists some pathogenic microorganisms that produce a cell capsule. And there were several mentioned in the lab module. There may be a table of it as well in the lab module. And then after those questions, answer 2 case studies. In case study 1, 45-year-old female is experiencing some respiratory issues. Based on her clinical presentation, the doctor orders a chest X-ray, shown here. And you can see something's odd about the lungs here and here. The lungs should be dark staining like this area here. And that's obviously very light So is that region there. Based on the x-rays, a sputum for acid-fast bacteria culture is made. There's the sputum. The cells are then stained with Zeal-Nelson stain. And then based on the findings below, what is the most probable diagnosis? You must name the species causing the infection. I don't want just the name of the disease. I want the species of bacteria that is causing the infection in this patient. Any questions about any of that? All right, in case study 2, we have a 64-year-old diabetic patient administered to the hospital with soft tissue necrosis. A routine culture and gram stain is ordered. The bacteria only grow anaerobically. Based on the findings below, what is the most probable diagnosis? Remember, you must name the species causing the infection. Can't just say the disease name. And there's the foot of the patient. That looks really bad. There's the bacteria that came out of the patient's foot. And that's probably his foot cells right there. And there's the bacteria. You should be able to tell me, this is the gram stain, what the bacteria are, their shape, and their gram stain. And then this is the Schaefer-Fulton stain. It is used to stain something, and I can see it in here. You have to find out what this is staining and what it is used for, and then answer the question. Name the species causing the infection. Now remember, do not write your answer in the lab module. I have the worksheet for the lab module. Well, I thought I had it right here. It looks like it's closed down. Let me go get the worksheet again. So write your answer only on the laboratory worksheet. It's a smaller page. If you submit the lab module, you'll get a penalty. The reason being is if everyone submits the lab module, my Canvas webpage will fill up, and then you guys won't be able to turn in assignments. So right here or right below the picture, give me the species which is causing the infection. And like I stated, the species is mentioned in the lab module. All right, any questions? If not, I'll be here until 8 o'clock to help you with any questions. But after 8 o'clock, or the last student logs out, I'm going to log out. So get started on the lab. I'll be here to help you.